Hello, everyone. I'm Manisha Deka, and I'm the Academic Director of the Animals in Society Research Initiative. Welcome to everyone joining us today online for the first presentation this academic year and what is now our 5th year running of the Animals in Society Research Initiative Distinguished Lecture Series. We're delighted to have you with us today. For information on other lectures in this year's series, please do visit our website, and Holly right now is posting the link on the chat for you, as well as an email address for our organization, the Animal Society Research Initiative, if you'd like to join our listserv and stay up to date with bi-monthly emails on news and events. Today, we are very pleased to welcome Dr. Aysha Akhtar, presenting under the title, How Can We End Animal Testing and Better Human Health? Dr. Akhtar is a double board certified neurologist and preventative medicine specialist with a background in public health. She's also a U.S. veteran and served as Deputy Director of the U.S. Army Traumatic Brain Injury Program, developing the Army's brain injury prevention and treatment strategies for soldiers. As a commander in the U.S. Public Health Service Commission Corps, Dr. Akhtar frequently deployed to assist with national public health emergencies. Dr. Akhtar was also a medical officer at the Food and Drug Administration for a decade, most recently in the Office of Counterterrorism and Emerging Threats, implementing studies on vaccine effectiveness and safety and using her top secret security clearance to develop national preparedness strategies for public health threats. She's published in peer-reviewed journals, including Lancet, Pediatrics, Journal of Public Health and Policy, and reviews in the neurosciences. A TEDx speaker and fellow of the Oxford Center for Animal Ethics, Dr. Akhtar is also author of Our Symphony with Animals on Health, Empathy, and Our Shared Destinies, published in 2019, and Animals in Public Health, published in 2016. We will have time following her presentation for a question and answer session. So we ask that you please post your questions in the chat at that time, and I will order those and read them out for Dr. Akhtar's response. Without further ado, let's welcome Dr. Akhtar.
Hi, everyone. Thank you so much for having me here. So what I'm going to do, since I know this is kind of a broad audience, I'm going to give a bit of an overview of animal testing, a little bit about its ethical issues, the scientific limitations with animal testing, and what the future holds in the world of biomedical research. So I'm just going to try to present my slides. All right, so the title is How Can We End Animal Testing and Better Human Health? So when we think about what animal testing is and how many animals are used, we can only use an estimate because there is so little transparency globally in many nations, and definitely in the United States, in exactly how many animals are used, how they're used, and how they are treated in laboratories. So the best we can do is estimate, and the latest estimate is about 200 million animals are used globally each year. In the US, that estimate is more than 100 million. So US is definitely one of the top nations for animal testing. And I will just say that these are likely underestimations based on the limited information we have. It very well could be that there are many more animals than what we're estimating here are used in experimentation globally and in the US. And the top 3 animal testing nations are US, Japan, which actually surprised me when I learned this, and China. So a lot of people don't have just a few bits of information to know why animal testing is done. So most people, when they think about animal testing, why is it done? They think it's used to test for drugs. It's used to test for the safety and effectiveness for drugs and vaccines. That category is what we would refer to as regulatory testing. Now, because I mentioned that there's so little transparency in the United States, we don't count the majority of animals that are used in experimentation, and we don't know how they're used. The best we can do to try to understand how they are used in the United States is to extrapolate based on the information in the UK, where there is more transparency. The UK counts all animals used in experimentation and categorizes under what type of basic category our animals use. And so there's no reason to think that the percentage of overall testing, the basic information that you see here, does not apply to the United States. So in 2018, the UK estimated that about 17% of animals are used in what's called applied research, and that's research that's really to hone in on specific diseases, for example, and to try to understand how those diseases manifest and how to prevent those diseases. Regulatory research is where most people think of research. This is in which there are procedures and testing is done on animals to meet legal and regulatory requirements by certain bodies, governmental bodies. In the US, the main two regulatory agencies are the Environmental Protection Agency, which regulates the testing of chemicals for toxicity, including environmental chemicals, and the Food and Drug Administration, or the FDA, which regulates the testing of drugs, vaccines, medical devices that may go in the body, and what's called biologics. So these are other types of, they're not quite drugs, but they're biological therapeutics that are used and vaccines are under that category of biologics. So what's important to note here is that most animals are not used for applied or regulatory research. Most used animals are actually used in what's considered basic research. And this is the type of research that often happens at academic centers. In the US, it's heavily funded by the National Institutes of Health, the main governing body in the US that funds biomedical research. And it's research that doesn't necessarily have a real world application or any kind of application. So it's research that we call like curiosity driven research, and it could be anything. It could be trying to understand anxiety in mice by having them do these tests in which they're forced to swim in a tub of water continuously to survive. And if they don't swim, if they give up, they'll drown. And this is called learned helplessness. It's basically research to understand how depression, anxiety, and learned helplessness occurs in mice. Now, the goal, of course, is to try to understand how it occurs in humans. But I don't know if many humans having to be forced to swim in a tub of water until they have to give up and drown. But there's also research that was done here in the United States that was funded by our government in the vein of, there was one research in the Northwest in which researchers captured songbirds from the wild, brought them into the laboratory, scrambled a part of their brain to understand how it affected their singing. They then killed the animals at the end and to see what morphological changes occurred in the brain. Now, in basic research, many researchers that carry out these types of experiments on animals will justify it in their publications by saying things like, this research may lead to greater understanding of Parkinson's disease, Alzheimer's disease, multiple sclerosis, and they'll often list a litany of diseases that this type of research may improve our understanding of. And that's because there's been increased public scrutiny of these kinds of research testing on animals, because there is no tangible real world benefit or not implied and immediate tangible real world benefit that's intended to come from this type of research. So in the US, what we do know is we have a count of the number of animals that are used that are covered under the Animal Welfare Act. And I'll go into that a little bit more. So what we know basically from the 2018 breakdown of the species used that are covered under the Animal Welfare Act, that you can see about 60,000 dogs were used in 2018, about 50,000 pigs, 70,000 non-human primates, a lot of rabbits, guinea pigs, and hamsters are used. From the 2018 statistics, from those animals that are covered under the Animal Welfare Act, 310,000 were identified as undergoing testing procedures that involved pain. And of those, 60,000 were identified by the research institutions themselves as undergoing painful procedures, but for which no pain alleviation was provided at all. And so I want to get to the Animal Welfare Act. It's the only federal regulation, federal statute in the United States that regulates the treatment of animals used in experimentation. There's a couple of things to know. One is that it excludes the majority of animals used in experimentation in the United States. It excludes more than 95% of animals used in experimentation. It excludes mice, rats, birds, and cold-blooded animals, invertebrates and fish and other animals of that vein. Those animals were intentionally excluded from the definition of animal under the Animal Welfare Act. The other thing to note is that for the animals that are covered, the Animal Welfare Act is basically provides a checklist. So laboratories have to check off certain boxes. So they have to check off that they're providing food and water for dogs and the animals covered under the Animal Welfare Act, and that they are providing basic housing requirements that may be stipulated for certain animals. What's important to note, though, is that the Animal Welfare Act does not at any point limit the amount of suffering an animal can experience. So the Animal Welfare Act, by statute, provides no limits on the type of experimentation that can be done to animals in the name of research, no matter how much pain and suffering it may cause to those animals. If we look at public opinion about animal testing, this is a poll. This is a Gallup poll that's conducted in the United States. It's been conducted since, if not 2001, even further back. And they asked the public several different questions. It's their morality and opinion poll. And so they'll ask the public questions like, how do you feel about abortion? How do you feel about using stem cells? How do you feel, in research? How do you feel about wearing fur? Things like that. And one of the questions they consistently ask is, do you find medical research on animals to be morally acceptable or morally wrong? So one thing I wanted to point out is that if you look at the trends from 2001 up to 2019, you will see that increasingly the public, there's an increased public disapproval of animal testing, medical testing on animals. Up to 2019, 44% of the public in the United States stated or checked off that medical testing on animals was morally wrong. Now you'll notice that there was a dip in 2020. And what I suspect is this dip occurred as a result of the pandemic and a result of many articles that came out that basically jumped on the pandemic to continue arguing for the use of animals in experimentation. In the United States, there was one huge article in the New York Times in the spring in which researchers who use non-human primates were arguing for greater funding for the use of primates in research and the breeding of primates, non-human primates, to create what they called a monkey stockpile. So there are articles like this that have been coming out in the past several years since the beginning of the pandemic, articles that are saying, we basically promoting the use of animals in experimentation and suggesting that if we don't use animals in experimentation, we're going to take a real step backwards in science. So I think these kinds of articles, in addition to the pandemic itself, led to that dip that we saw in the public poll. What I suspect is that that dip is only temporary, and once this pandemic subsides, that poll will continue to see that rising trend in disapproval that we saw. One of the things we also saw with the pandemic is that there's been a strong case to make that we moved so quickly in developing and improving vaccines for the pandemic, in part because the traditional tests that would normally be done on animals for vaccine development did not occur. Meaning, and it's not to say that no animal tests occurred, but normally in vaccine development, animal tests will be done before a vaccine is tried in humans. Because of the public health threat that we face, that kind of paradigm was shifted. So the sometimes months long, years long animal testing that may be done for vaccine development was bypassed so that they occurred some of them occurred simultaneously with the human clinical trials. So that really, when you think about it, humans are the gold standard for human biology. And so when we're talking about using animals, when we're already at the human clinical trial phase, it suggests that we do it, that the institutions do it because we're afraid in some ways to fully deviate from that protocol. But it really begs the question, why did we even do the animal testing when we were already at the human clinical trial? But nevertheless, it's been strongly argued that the vaccine development process occurred very rapidly, in large part because a lot of resources went to it, but also because many of the normally the animal tests that can take quite some time were bypassed or conducted concurrently with the human clinical trials. So I'm a neurologist. I've been a deputy director of the traumatic brain injury program in the Army. And we had to, basically my role was to help develop detection, prevention, and treatment strategies for soldiers with head injury. And as you can imagine, head injury in soldiers is a big issue. The truth is we don't have any treatments for head injuries itself. We have treatments for some of the symptoms that result from head injury, but we have no treatment for the head injury itself. We also, as a neurologist, I can't tell you how many times I've had to diagnose patients, even friends, with diseases like Parkinson's disease, Alzheimer's disease, and to have to tell them that there are not a lot of treatment options for those diseases. And again, the treatment options for most of those diseases, don't actually treat the disease themselves. They help with some of the symptoms that can result from the disease. And so this is a picture of my father, my Abu, drinking his morning cup of tea. And he suffers from diabetes. And as a result of his diabetes, he frequently experiences a kind of pain called neuropathy. And if any of you have experienced this kind of pain or know someone who has experienced this kind of pain, you will know that it's a very different kind of pain from the traditional pain we might get, like a cut on the finger or something. It's a very excruciating, uncomfortable pain. And it's a pain that is very, very difficult to treat. I have seen my father suffer time and again from neuropathy, which frequently occurs as a result of diabetes, and not be able to offer him any treatment that really touches his pain in any significant way. And so my experience with my father, my experience as a neurologist, my experience at the Army, and then my experience at the Food and Drug Administration for 10 years, in which we saw drugs and vaccines come through the pipeline again and again, in which there was tremendous promise and hope about those drugs and vaccines, only to see most of them fail when tried in humans. Those experiences had led me to kind of reframe the ethics, to not ask, just is animal experimentation ethical for animals, but is it even as a whole ethical for humans? So to answer that question, we have to ask these other two questions. Is animal testing an effective use of resources, and does it reliably inform human health? Now, I'm not going to go into all the evidence here because I know this isn't a predominantly scientific community. But if you want to get some more information about this, you can definitely reach out to me. But I'm just going to give you just a couple of examples. So we talked about basic research, in which that's the area where most animals are used in experimentation. In the United States in 2000, all the way back in 2000, the Institute of Medicine, which is a pretty influential medical institution here in the United States, convened what they call a clinical roundtable to assess the how well basic research is translating to actually human benefit. And so what they came out with, the report was, and I will say they didn't look specifically at animal research as part of basic research, but basic research as a whole, which includes a lot of animal research, if not most animal research. And what the clinical roundtable basically reported from their assessment was that despite the immense resources that go into basic research, there is a strong disconnect between the immense resources that go into basic research, the promise of basic research, and actual tangible human benefit, meaning that basic research as a whole, despite the immense resources that go into it, does not really lead to effectively and efficiently lead to tangible human benefit. Now, if we look at drug development, which is the second big category of animal testing, in the United States, and this has been, you might have heard this statistic, in the United States, all new drugs and vaccines, with rare exception, per FDA's regulations, must be tested on at least 2 different species before they can move on to human clinical trials. So they must be tested usually for safety, definitely for safety, and for effectiveness in at least 2 different species. Usually one species is a ferret, a rabbit, a mouse, for example. The second species will often be dogs. Dogs are in the United States one of the major animals used for drug development. They may be non-human primates, or increasingly they are using pigs. And so about 10 to 15 years ago, the Food and Drug Administration, the Health and Human Services in the United States stated that about 90 to 95% of the drugs and vaccines that come through the pipeline fail to make it to the market. And what that means is that 90 to 95% of the drugs and vaccines that pass the animal tests, that are found safe and effective in the animal tests, as well as other tests, other types of in vitro cell culture tests. But because animal tests are considered pivotal for decision-making by the FDA, they are crucial in this process. So 90 to 95% of the drugs and vaccines that pass the animal tests end up failing in humans when they're tried in humans. Most of them fail because they're found to be unsafe or ineffective when tried in humans. So think about that just for a moment. 90 to 95% failure rate in drug development. Do you know of any other industry that accepts such a high failure rate? I mean, can you imagine if you hopped on a plane, you start to buckle in, and your pilot says, all right, ladies and gentlemen, strap down, because I will tell you that we have less than a 5% chance of landing safely at our destination. I mean, we'd all hop right back off the plane and demand an overhaul of the entire airline industry. But for some reason, this failure rate has been accepted in drug development and vaccine development. And so there have been several reasons offered for this high failure rate in drug development. And this was based on an article in Drug Discovery, came out by pharmaceutical experts. One of the potential reasons was that there are potentially higher regulatory hurdles, meaning that regulatory agencies like the FDA are becoming more and more demanding of what the criteria for a drug to be approved for the market. That certainly, there's certainly some point to that. Clinical trials are long and expensive, and that we do know. Running clinical trials with human participants is long, it's difficult, it's expensive. It's difficult often to recruit people to participate in clinical trials, and it's difficult to keep them enrolled in the clinical trial. So there's a high attrition rate. So that's definitely a reason for this high failure rate. But one of the main points that came out from this article was that the poor predictability of animal experiments was a significant barrier and a significant contributor to this high failure rate in drug development and vaccine development. So it's, we've been doing animal research for centuries, really. And, when we think about it, we started using animals for medical research because that was what we knew. And initially, when a lot of research was being done, there were many things that were translatable between species. So you could take a look at one species and study some basic biological mechanisms, and you can see that that's also something that's occurring in humans. But we've really moved on in medical research, and yet we've continued. We've become more sophisticated in medical research and what we demand of medical research, yet we continue to use basically a tool, animals, that has been around for a long time without really asking if it's really very effective. In 2007, a groundbreaking article was published in the British Medical Journal. And this led to a lot of controversy and a lot of dispute, especially by those, I suspect one of our listeners here would be among that audience, but especially by those who felt that, who basically were not happy with what the article was suggesting. But basically, so what researchers did, some researchers decided to see how effective and how predictive is animal research in telling us what we're going to see in humans. So what the researchers did is that they looked at 6 different treatments across 5 different disease categories. And this included respiratory distress syndrome, osteoporosis, stroke, hemorrhage, and head injury. And what they did was they gathered, they looked at all the published animal data on those treatments and all the human clinical data on those treatments, and they compared the results to see if they match. And what they found was that the animal data matched the human data only 50% of the time. In other words, the animal research was no more likely to predict whether those treatments were going to be safe and effective in humans than a toss of the coin. So since this was the first, I think this was the first of its kind that really took a good, what we call a systemic look at animal research. And since then, there have been numerous articles that have come out in the same vein that really do suggest that there is very limited predictability of animal research for human results and human outcomes. And so if I just talk a little bit about some of the ways animals have used, if we look at stroke experiments, and I mentioned strokes, there have been more than 700 treatments that have been studied in animals in the lab and have been found effective to reduce the effects of stroke in animal experiments. Of these, more than 150 were tested in human clinical trials, yet not one proved effective in humans. If I give you another example, for decades, researchers have been trying to find an HIV vaccine that will help prevent HIV. And so HIV vaccine has been tested in a number of animals, including specifically non-human primates. And for a long time, they were also tested in chimpanzees until the use of chimpanzees were pretty much in disfavor in the United States, which occurred about 8 years ago. So there have been at least 85 vaccines that were found effective in animal experiments in preventing HIV. Of these, 30 were tested in human clinical trials, and not one proved effective in humans. As a matter of fact, 2 of the vaccines actually showed increased risk of HIV in humans, meaning the vaccines increased the risk of HIV in human participants. there was just recently another HIV vaccine that people thought held an immense amount of promise. There was a great deal of excitement about this HIV vaccine. It was tested in animals again. It worked in animals. The U.S. has spent hundreds of millions of dollars in developing this vaccine and testing, and yet again, it fails when tried in humans. So if we look at the drug development, so what I showed you were the failure when something is tried in humans. So the lack of prediction of animal tests in suggesting whether treatment is going to be effective and safe in humans, and how that can lead to immense failures in clinical trials. I'd like to take the opposite view. So this is the average drug development timeline in the United States. It takes about 10 to 15 years for a new drug to go from the drug discovery phase, the initial phase in which pharmaceutical companies may be looking at thousands of compounds to see if one might be something that could be useful against a certain disease. From that initial phase all the way to the market, it takes on average 10 to 15 years for those drugs and vaccines that do make it to the market. So you can see it starts with about 10,000 compounds on average, then about 250 of those compounds are tested in preclinical tests, and about 10 may move on to human clinical trials. And as we know, 1 or less than 1 out of 10 will actually be approved. What I want to point out is the section between the preclinical and the clinical. So in this regard, preclinical means before human testing. Clinical trials means human testing. So preclinical includes animal testing. It includes computer models. It includes cell cultures. But what I wanted to point out is, again, animal testing in the United States, and I believe this is globally, there's no reason to believe otherwise, is considered crucial by regulatory agencies in determining whether a drug or vaccine should move on to clinical trials. So here we see about 250 compounds on average may be tested in the preclinical phase, so in the animal testing phase. And out of those 250, only 10 may be tried in humans. What happened to the 240 compounds that didn't move on to clinical trials? And increasingly, people have been asking how many of those 240 compounds would actually have been of benefit to humans, would have been safe and effective in humans had they been allowed to move on to clinical trials, had they not been tested on animals, and had they been allowed to move on to clinical trials. So what we're talking about here is that because of the poor predictability of animal testing in telling us whether a drug is going to work and be safe in humans, there is a high likelihood that drugs and vaccines that would have worked in humans, would have been safe in humans, were actually abandoned based on the animal tests. So how many drugs that would have been effective were abandoned because of misleading animal testing? And so we do have some examples of near misses in this process. So for example, Gleevec is a drug that is used to treat a form of chronic leukemia. It showed severe toxicity, liver toxicity to such a high degree in 5 different species that it was almost abandoned and not moved on to human clinical trials. Now, although liver toxicity is still a risk factor for humans, it's not a risk factor anywhere near to the degree that it was in those animal tests that were done. And fortunately, the drug developers, when they were using different tests, also found that the compound did not cause significant liver toxicity when they used human cell assays. So because of that, they moved on. They were allowed to move on to human clinical trials. Unfortunately, we have the drug. Tamoxifen, there's a drug called Tamoxifen that's used for breast cancer. There's a drug called cyclosporine that's heavily used to prevent organ transplant rejection and used to treat a variety of autoimmune disease that were almost abandoned because of test results in animals that were not applicable to humans. So just a summary of the drug development pipeline. It takes about 10 to 15 years from a candidate drug to go from the investigation, from the lab phase to the market, if it does make it to the market. It costs a drug developer, so a pharmaceutical company, on average, 1.8 to 6 billion US dollars. Now, I don't know about you, but I personally don't really care how much a pharmaceutical company spends to develop a drug. I don't have a lot of sympathy for the Pfizers and the GlaxoSmithKlines in the world. However, the problem with this high drug development cost is that this high cost is then passed on to the consumers in the form of high drug costs. So we actually do pay for this high failure rate and the high drug development costs that pharmaceutical companies endure. And then, as we talked about, the average success rate for a new drug or vaccine to make it to the market in the United States is only about 5 to 10%. So if we were to kind of summarize some of the human harms that can come about by using animal testing at this stage in where we are in biomedical research, meaning at this stage in sophistication and need for the types of testing methods and the types of answers we need when we conduct medical research. We can talk about three types of human harms that result from this. One is that there have been multiple examples of adverse events, so serious, many people think of these as side effects, but serious safety problems that occurred in humans that were not predicted by the animal tests. Two, we talked about a little bit about the immense time, money, and resources that just go into the drug development process. We're not even talking about basic research. We're just talking about the drug development process, only for most drugs to fail when tried in humans. So there's an increasing argument that there's an ineffective use of time, money, and resources by being so wedded to animal testing. And then three, a very likely harm is the missed opportunities, meaning we might have missed drugs that could have worked, and vaccines that could have worked in humans, but did not go on through the drug development pipeline because of effects that were specific to the animals, the species that they were tested on. So if we don't do animal research, or what can we do instead? The good news is that over the past 10 to 15 years, there's been some significant technological breakthroughs that have come about in medical research. One of the biggest technological breakthroughs, and something I don't think anyone would have thought of 15 years ago, is what's called human organs on a chip. And basically what this is that you can grow, often an institution will take adult stem cells, reprogram them into different types of cells. So they may reprogram them into human kidney cells, human liver cells, human lung cells, and then create a three-dimensional structure, three-dimensional culture of these cells in what's called on a microfluidic device. And I'm not going to get into the specifics, but basically these are recapitulations of a significant part of an organ that is distilled onto a small microchip. So for example, we have the human lung on a chip, we have the human kidney on a chip, we have the human gut, human heart, human mini brain, we have the human blood vessels on a chip, we have the bone marrow on a chip, and so forth. And in many cases, these different chips are actually connected to create the human body on a chip. So these allow for more whole body physiological testing. There are other types of testing methods that have come about, such as organoids, and these are a little bit similar to the three-dimensional organ on a chip, but these are a little bit more, even more, a further step in a sense, that they are miniature organs, basically, that are bioprinted in the lab. We are creating virtual humans based on the incredible array, incredible data we have on human biology. And I won't go into these other types of projects, but these are some of the types of testing methods that are coming about. Now, I will say, and there's smart AI now also being used to create what's called virtual humans. Now, I will say that no one of these tests is perfect by any means. There is always room for improvement. There's complexities in doing this, running these types of tests, and they don't capture all the information we need. And no one testing method is going to give us all the information we need. What's important to note is that it will take a variety of testing methods to give us the best picture of the human body, of human physiology. So it will take testing methods like virtual humans, in silico, computer modeling, organoids, three-dimensional cell cultures, MRI imaging, clinical trials, and so on, to really get a good understanding of human biology and human diseases. One of the exciting things that's happening now as a result of these type of new innovations that have happened is that we are going to be moving into the era of what's called personalized medicine. So I know many of you have, especially during this pandemic, we've seen a lot of articles about why there is a need to capture diversity in clinical trials. So we need to capture people from diverse age ranges, diverse backgrounds in clinical trials. Traditionally, clinical trials historically were predominantly made up of Caucasian males. And now we know that that's not enough. That doesn't give us a full picture of how a drug or vaccine may work in the entire human or in the larger human population. Because what may be safe and effective in you may not be safe and effective in me, may not be safe and effective in someone like my mom, or safe and effective in my Caucasian 50-something-year-old husband. So we know that one person's biology may not predict what we'll see in another individual. So the beauty about these type of testing methods that have come about is that we can actually capture that diversity in these types of testing methods. So researchers can use my cells to create a mini Aysha or an Aysha on a chip, or capture Holly's cells to create a Holly on a chip, or my husband's cells to capture a Patrick on a chip. And so we can actually test for how a drug or a vaccine may work in different populations and different categories within the human population. The next thing that leads to personalized medicine. So what I mean by that is that eventually, let's say I have a disease, I have a cancer, I develop a cancer. So eventually, where we will go, is that my cancer cells, my cells, will be used and grown in the lab, and drugs, or in this case, drugs, will be tested against my cells, against my tissues, not against Holly's tissues, not against some random person in the population's tissues, but against my tissues to see if that drug will be effective against my cancer. So that's called personalized medicine. And that's where we're heading. And that's where we can head because of these great new technological advancements that have occurred in recent years. And the last thing I'll say about these testing methods is that we very well soon will be conducting clinical trials in the lab. So we talked about clinical trials. That's when people enroll to participate in a test for a drug or a vaccine. But we can actually, with these types of testing methods, because we can capture diversity in the population, we will eventually be able to run clinical trials using these types of testing methods. So we can get a lot of knowledge about the safety and effectiveness of drugs and vaccines before they're actually tried in living humans. And the important thing to remember is that these testing methods are based on human biology. So there's no guesswork in regards to whether the findings from one species is going to be applicable to what we see in humans. There may be other types of guesswork, but there's not that species different guesswork here in this process. So there have been a lot of signs of change in biomedical research. In addition to the technological advancements that I've been talking about, there have been some major changes that have occurred. And of course, because I'm in the United States, I'm going to mention what's happening in the United States. So in 2019, the Environmental Protection Agency, which regulates chemical testing, environmental chemical testing, declared a memorandum that is going to end all mammalian, not all animals, but the testing on all mammals by 2035. Meaning chemical companies that by 2035 will have to use more human relevant non-animal testing methods. So there's been a rapid increase in innovation in this area as a result of this mandate. You know, when there's a need, you know, when necessity is the mother of invention. There's a necessity here for chemical companies to be able to reach, to be able to meet this mandate in 2035. And lo and behold, they are finding new technologies, new testing methods that not only replace animal testing, but they're better, they're more predictive. They can be used faster and they can be used more cheaply. In the United States, multiple states have enacted bills to outlaw the sale of any cosmetic that's tested on animals. There's an analysis done by Research and Markets last year, which looked at the market for animal testing versus non-animal alternatives. So non-animal alternatives, they looked at organs on a chip, organoids, in silico, and other types of testing methods. And although currently the global market for animal testing is much higher than the global market for these non-animal testing methods, the market, the projection is different. So what I want to point out is that but the projection for the market for animal testing, global projection from 2019 to 2025 is 4.2%. Then from 2025 to 2035, the market projection for animal testing actually decreases, goes down to 2.4%, 4.6%. In the United States, you can see a similar dip in the market for animal testing. Alternatively, if you look at the market for these human specific testing methods, the market for both globally and in the United States is much higher. As far as the growth rate is much higher for these non-animal testing methods than they are for animal testing. So what this suggests is that the market for animal testing is plateauing and then it's going to take a dip. However, the market for human biology-based testing methods is increasing and increasing at a much faster rate. So that's the good news because, and the reason why they're suggesting that is because of articles like this that you see, the next phase of drug creation, why models are on the brink of extinction. It is widely known now that animal testing is so poor in predicting human outcomes, especially for drug development and vaccine development. It is so widely known. Pharmaceutical companies, there's Pfizer representatives in this article talking about we need to move away from animal testing. There's a large publication came out by a major UK pharma industry consortium that said we need to move away from animal testing and use more human relevant testing methods. What they're suggesting is that we have probably getting close to reaching the limit of what we can learn from animal testing. And we're getting to that limit because we're now looking at the subtle nuances of molecular biology and genetics and physiology. And in this area, even small differences between species can have a significant result in the outcome, in a study outcome. So it can significantly impact the study outcome. So we really need models that are based on human biology, not the biology of a rat, not the biology of a dog, not the biology of a cat or a non-human primate or a pig or a ferret. We need models, we need methods that are based on human biology. We at the Center for Contemporary Sciences also did a pilot study of students' preferences in training. And so what we did is we conducted a pilot study of biomedical students obtaining their PhDs. And we gauged how interested they were in learning about human-specific testing methods. And of course, 97, the majority, said they were very much interested in, or they were interested in training and learning about human-specific testing methods. the reasons cited, and they were able to pick more than one. The number one reason actually cited was ethics. They were concerned about animal suffering with animal testing. The second major reason cited is that they felt that these methods were more human relevant. And oh, I see I have a typo here. I apologize for that. And then the third reason for their interest in human relevant testing methods is that they felt that human-based research is the future of science. However, despite this interest in learning about these newer, these more human-specific testing methods, half of the students said that there were no opportunities, or they were not being made aware of any opportunities to train and to learn about this human-specific research methods at their universities. So what that really suggests is that there is an unmet need at the university curriculum level, that they are not preparing students for where the market is going, actually, for where the market is going towards more human relevant testing methods. And so there are several barriers. Despite what I mentioned, there are several barriers to replacing animal testing. One I mentioned is education. So it's not just education of the emerging scientists, but also education for many scientists who have been out there for years. They've done animal testing. They're not familiar with these new testing methods, and it would take education and training to learn how to use these new testing methods and how to apply them in their own line of research. There's policy barriers. Currently in the United States, as I mentioned, the FDA, despite the immense failure rate in animal testing and predicting whether a drug or vaccine is going to work in humans, the FDA still demands, with rare exception, that all drug and vaccine sponsors must test their new drugs and vaccines on at least 2 different species. So it's policy changes like that that impede innovation, impede the use of these other potentially more effective human relevant testing methods. There's a cultural barrier. Many folks, they have been trained to use animals in their academic careers. It's what they know. It's what they're used to, and so there's always a difficulty in overcoming that cultural barrier. And then the last thing, and I will say this is probably the biggest barrier, is money. There is very little money in the United States funding, governmental funding, that goes towards the development and improvement and use of more human relevant testing methods in comparison with the amount of funding that goes towards animal testing. And that needs to be switched. So we are at the Center for Contemporary Sciences. We're a new organization. We launched just last year at the beginning of the pandemic. Yay, a great time to launch a new nonprofit organization. But we're basically, our role here is to help create a worldwide transition to using more human-specific medical research that will not only reduce and replace animal testing, but that can offer even more benefits for human health. And we do this by educating the next generation. We plan on creating curriculum for students and establish career scientists on how to use these new tools that are coming about. We are working on policy changes here in the United States to allow for more options to be used in drug development and vaccine testing. So it's policy changes that don't take away animal testing, but allow drug companies to use these other types of testing methods if they feel that they are going to be more predictive of human outcomes than the animal testing. We need to change the narrative, and we should change the narrative. The narrative traditionally has been that it's your dog or your child, meaning you have to choose between saving the life of a dog or saving the life of your child. So it pits animal welfare against human health. But that narrative needs to change because the science is showing that it needs to change. What we're increasingly finding is that if we want to improve human health, we have to move away from animal testing. We have to start embracing these other types of testing methods and putting more funding and investment in who knows what the next big breakthrough in innovative testing will be to improve human health. So there's a win-win benefit moving more towards these other types of testing methods. And the other solution that we are also working at the Center for Contemporary Sciences is to increase investment and governmental funding towards the development, the improvement, the refinement, and the use of these more human relevant testing methods. So with that, I'll just stop there. This is my Nessie. I gave a picture of my dad, so I wanted to balance it also with a picture of my Nessie, my little feline, and that's her favorite soccer ball. So I'll just stop sharing, and we can open up to Q&A. Let's see if I can do
Great.
this. Okay.
Thank you so much, Aysha, for that excellent presentation. Yes, we now have time for question and answers, and we have a first question in the chat from Jay, who asks, how do we replace the basic category of animal testing, and how do we motivate people who receive funding for these studies, especially those that cause suffering, pain, to reorient their work?
Yeah, so what I suspect is that it might be difficult for those who, are really used to using animals. It might be difficult, very difficult to kind of get them to switch. But one way to move them in the other direction is by increasing funding towards that direction. So I'll give you an example. So I've spoken with many academic centers that are creating like human organs on a chip, and they apply for funding, governmental funding from the National Institutes of Health. And what so many of them have told me is that when they apply for governmental grants and funding, they get kickback from the NIH. They're being told, why aren't you doing animal testing instead? So that needs to change. That prioritization of animal testing versus other types of testing methods need to change. And so it's important to change how our governmental organizations fund medical research. If we can change that, the research will change. People will go where the funding goes, right? Researchers will go where the money is ultimately. If the funding is more towards human relevant testing methods, more researchers will be moving in that direction. And as far as basic research, so a lot of these testing methods are being used in many areas of basic research to study basic biological mechanisms. So lung on a chip, there's one lung on a chip that's created by the Wyss Institute at Harvard, and Emulate, which is a private company that is being used by the government here in the United States to look at how the coronavirus affects our lung tissues. So it is being used for that type of basic research, which normally would be done in animals. And it's also being used for more applied research, and that it's being used to screen drugs for their effectiveness in treating or preventing coronavirus. There are some types of basic research that there's no replacement in a sense, because there's a lot of psychological behavioral experimentation on animals. Organs on a chip isn't going to change, isn't going to replace that. What's ultimately going to replace those type of testing methods is really the understanding that those type of psychological behavioral experiments on animals may not be the best use of resources. They don't really give us good insight into the complexities of human mental illness, human psychological disorders, autism, things like that. So there's a different mechanism to try to change that type of research that is done on animals.
Okay. And if I could take you back to just one of your last slides where you're talking about the markets for human versus animal. Yes. Please excuse my ignorance, but what exactly is like the market for animal research? Like, can you describe what that is? So what does that 4.27% figure represent?
So we're looking at, so there are a lot of companies, a lot of private companies, there's a lot of businesses involved in biomedical research. So there are many that are involved in animal research. So there are what's called contract research organizations in the United States, and many of these breed and house animals, and other institutions will contract out with those companies to conduct the animal testing on their behalf. So the models they create in for like, they may create, a so-called rat model of Parkinson's disease, which really isn't a model of Parkinson's disease, but may capture some symptoms, but they'll have a rat model of Parkinson's disease and things like that. There is market, how do I explain this? There is a business goal with creating these types of models. And so there's a economic advantage in a sense for some. There's a business advantage for some companies to use animal testing in that way. I know I'm butchering my explanation here, and forgive me. I'm not an economist, but I'm trying to capture this information. On the other hand, the non-animal testing methods, there are a lot of private companies now, and academic centers and governmental centers that are paying to use these human organ on a chip technologies, organoids, virtual human testing methods. So I hope that makes sense. So the growth in the economic market for non-animal testing methods is growing at a much faster rate, and the economic market for animal testing is actually plateauing, and it's projected, we'll see, it's projected to decrease over time. I know I didn't answer that sufficiently, and I apologize.
No, that's fine. That's fine. And so just a reminder that if you have a question, to please post it in the chat. Aysha, can I ask you, because of course my background is like regulatory, being immersed in the law school, and I did not know about the EPA and the mammalian goal for 2035. And so we were speaking also that like some of the appetite for change is coming from those who just see that there's not much more to learn from animal testing because of the subtle molecular differences. So is it more like, what is really driving a change at the EPA or other regulatory agencies that you may have noticed worldwide? Is it this, okay, we just can't get any more good results, or is it any sense of animal ethics, or is it both?
With the EPA mandate, there was a sense of animal ethics involved, but the real drive that's happening in replacement of animal testing is not an animal ethics drive or based drive. It is because there is a human health need for more predictive or human relevant testing models. In the United States, most chemicals that are actually on the market, most chemicals that are being used right now, that companies are using for agricultural products, petroleum products, and anything you can imagine, have actually not undergone any type of testing for safety at all. Part of the reason, so there is a huge backlog of testing of chemicals in the United States, and I believe that's also in the European Union and in the UK. And so part of the reason why there is this huge backlog is that animal testing can take quite some time to conduct. They're expensive, and they're not very reliable, meaning that they're not very predictive. So then, often you're left with, we don't know if we can trust this result, so we now have to do this other testing to see if this is going to be more likely to predict what we're going to see in humans. So the alternative methods that are coming out, they're what's called high throughput, meaning that you can screen chemicals through these different types of methods at such a fast rate, and actually at a rate that can basically, you can screen a number of chemicals at a rate that you cannot using traditional animal tests. So there's been a real need for testing these chemicals for which they have not undergone any kind of testing, and there's been an acknowledgement that animal testing is not being very helpful in this case, and that we need faster, cheaper, and more reliable testing methods. And that is really driving a big part of the change that's been happening at the EPA. There's also, within scientific communities, and I would say what I've talked about here, there is a growing momentum within the scientific community. Yes, animal testing is a predominant feature of biomedical research currently. But because of the limitations of animal testing, there is a growing momentum within the scientific community calling for changes in how we do biomedical research, calling for a move away, not immediate end to animal testing, but a move away from animal testing and replacement of animal testing with more reliable, more human-specific models. And so that momentum is really growing. And I suspect, we haven't reached a tipping point, but I suspect that we're going to reach that tipping point in the next 10 years, at least in the United States, possibly in Europe and the UK, maybe even in Canada.
Okay. Aysha, we have a lot of chat comments thanking you for such an excellent presentation. And Becca has sent a question as well. Thank you for a fantastic talk, Aysha. Have you worked at all on connecting the replication crisis in basic science to your efforts to end animal testing?
I have personally not worked on that. We have, at our organization, we have Dr. Carmen Kadir, who has a PhD in biomedical sciences. She's a basic and clinical researcher, and she will talk till you just want her to stop in some ways, because it's too much detail about how there is a replication difficulty in animal testing, meaning that the results from one laboratory don't always match the results from another laboratory. So there's been a big problem saying that animal testing, one study does not replicate what another study has produced, does not replicate the same data that another study has produced. And so because of that, there's been a call that we have to standardize animal testing, and meaning like use the same, not only the same species, but the same strain. So it's not enough just to use mice, but you have to use the same type of strain of mice across different testing methods. You need to ensure that they're being housed in the same way, they're being handled in the same way, that the research protocol is the same across different testing labs. There was actually a study, a really good study, that actually looked at whether or not you can standardize these methods across different laboratories to increase that replication. So they basically had looked at 3 different laboratories, very different laboratories throughout that were, I think one was, I have to go back, but I think 2 were in the Europe and one was in the US. And they standardized everything about a particular research study, everything that they could think of, standardized, meaning how often the animals were handled, how they were caged, things like the lighting in the laboratory, and then of course the research protocol itself. And what they found is, despite that attempt to standardize, the results widely differed across the 3 different laboratories, even for the same study that was implemented. So that is a major problem and a major limitation of animal testing. So, we might still face some of that problem, even with some of these more human relevant testing methods, because one organ on a chip, lung on a chip, produced by an emulate, for example, may not be the same as a human lung on a chip produced by another company. So there is a need to standardize these testing methods too, to ensure that there is replication across different types of studies. So there is an effort being undergone in the US to create standardizations for how you use these type of testing methods.
Okay. And another question is, do you recommend any current documentaries for general public education on the issues you've covered? There was like a 2012 documentary, Maximum Tolerated Dose, but are there more current titles that you recommend for anyone looking for a general overview?
There is a documentary that's being produced and that will be released by an organization, I think it's called Center for a Humane Economy. So look for that. I don't know when that's coming out. So I am in it and some other researchers are in it, but I don't know when that's coming out. So that's probably the most recent. And it's really looking at the science behind animal testing, not really so much on the animal ethics part of it.
Okay. Another question. how would we get funding bodies to value the organ on a chip methods over animal testing? Do we send them info or invite them to talks? And I guess I would just add my own addition to that question, like what has been the reception for your organization and for your message in the kind of the status quo or communities committed to the status quo?
Yeah, so there are many communities within the biomedical research space that are committed to the status quo. And, we'll try to reach out to see if there's potential for collaboration on, for example, we reached out to the Michael J. Fox Parkinson's Foundation to ask if they'd like to join us in in promoting a type of policy that could expand the type of methods that can be used for Parkinson's and other types of research. And the response immediately was, we believe that we must do animal testing and no other testing can replace animal testing. And we weren't saying that we were replacing animal testing. We were saying that we were looking to see how other types of testing methods could possibly be used. They may be able to replace animal testing in certain areas. I would argue they should replace animal testing across most areas. But it was a gradual change that we were suggesting just to try to collaborate with them. So when we get that kind of answer from folks, to be honest, I don't really try to work, try to break that wall with the institutions that are really wedded. I will die of frustration if I keep trying to change the people who are really wedded. Fortunately, as I mentioned before, there are so many academic centers, so many nonprofit organizations, so many people, even within the governmental agencies that I mentioned that were part of the problem, but individuals and offices within those agencies that are very forward-leaning in this regard, that are very much looking for how can we move the mark here? How can we move towards more human relevant testing? And so I work with those folks, I don't work with try too hard with the negative Neds and negative Nancys in the world here. I work with those who see what the future can hold and are embracing how technology can really drive innovation in medical research.
Thank you, Aysha. Another question, we have about 5 minutes left here. Thank you for all the questions, everyone. Do you think animal testing continues in part because of the profit-making industries that have involved around it?
I do think that is a big part of it. The reason why the majority of animals are excluded in the United States under the Animal Welfare Act is it was based on heavy lobbying on the part of groups that their sole purpose is to promote and maintain animal research. And part of their objective, part of the way they do that is to continually put out misinformation in the media and the public to convince the public that we absolutely need animal research. There's no way we can replace it. Other testing methods will not really be able to be very useful for us and blah, blah, blah. These were the same folks that had this big New York Times piece about why we need to create a monkey stockpile in the United States. So that's absolutely happening in the United States. And, I again, just for those folks, I think the work for us, for many of us, is to really show that narrative that they keep perpetuating is false. Is absolutely false. Unfortunately, it's the science is showing that, and the scientific community is starting to embrace this new narrative much more than it ever has before. So that public perception will change as the scientific momentum continues to build.
And do you think there is like a generational change, perhaps from your pilot study or otherwise, the United States? And also, do you think there is like a discernible regional, like international difference? Like I know India is doing very interesting work trying to lead the conversation away from animals. Like, do you see some standout countries or standout centers outside of, let's say, Canada, the United States with this work?
I think so. Some parts of Israel is making some, putting a lot of research and funding into newer human specific testing methods. And I have heard and seen about what's happening in India. There have been other countries. So is it Sweden? Sweden is a major, they've actually, their government is fully on board with an ultimate full replacement of animal testing. And I forgot exactly what their goal is, but their goal is to end all animal testing within a certain timeline. And so they are really working hard to change how biomedical research is conducted. And because, because there is a will there, they will find the way, they will find more testing methods that will not only replace animal testing, but actually will be much better.
Mm. Well, just on that note, as a closing comment, we had our first ever, we were having a federal election here in Canada, and one of our animal justice organizations called Animal Justice held the first ever kind of all leaders debate on animal protection. And so that's available for our audience on Animal Justice website. So I was listening to that and I thought I misheard, and even now, perhaps I did, I'll have to go check it. But one of the candidates said of the present government that there were internal caucus meetings about that, actually that policy, like ending all animal testing. And I just couldn't believe it that was like on the discussion table. So it was, very encouraging to hear if I didn't mishear. Yes.
That's amazing. And I think, honestly, the reason why we're seeing more and more of that type of sentiment being expressed is because of the amazing innovation that's been happening. And people are seeing what we can do if we put our mind to it, and, how much we can improve biomedical research without using animals.
Well, Aysha, I cannot thank you enough for, treating us today just to this wonderful presentation, so highly polished, and for answering so many questions with such patience and grace. We really, really appreciate it. And we hope that we can, invite you back at a future point, and love to stay in touch with your center. And if there's any thing you wanted to put in the chat for the website, feel free to do that. And
Okay.
Let me also thank all the audience members who are here with us today. Thank you for your contributions. So next up in our series, we have Dr. Kritika Srinivasan from the University of Edinburgh, the Senior Lecturer, School of Geosciences, and the title of her talk is The Ethics, Politics, and Ecologies of Farming Animals in Multicultural Societies, Insights from India. So once again, thank you, Dr. Akhtar, and thank you everyone in the audience for joining us today. And we hope to see you at our October series lecture.
Thank you so much. And I did add the link to the chat.
Great, thank you.
Thank you, everyone.